and in person. It's so funny to have this virtual hybrid model. Dr. Jessica Dewis, who's a pediatric geneticist at Children's Hospital Colorado. Really excited to have her here today, talking about the genetics of our diagnosis and how to help understand that.
Yeah, thank you all so much for having me. So I'm going to talk in general about genetics, and then there'll be some breakout sessions that will be specific to each specific syndrome. So these are some of our learning objectives. So I'm just going to, in general, talk about chromosomes, copy number variants, and then some of the mechanisms by which we see these occur. And then we'll talk about how genetics can be involved long-term in care. So when we do genetic testing, what we do is obtain a sample, which sometimes can be difficult. And what we do is we basically pause the chromosomes during their division in the cell, and we look at the chromosomes directly. So this is a structural way to look at the chromosomes called a karyotype. And what you can see from these two visuals here is that one of them is a girl with 2 X chromosomes, and one of them is a boy with 1 X and 1 Y. And we have 22 pairs of autosomes, and we're going to focus on the autosomes in this discussion. So the other type of testing that we do is a very specific test to look for small missing or extra pieces called a chromosomal microarray, which many of your children may have had. And when you look at those, this is how we look at this test. So we look at a reference and we look at the particular sample, and each color in the well tells us if there's any missing or extra copies. So you can see the green wells would indicate that there's an extra copy, and the red would indicate that there's a missing copy. And it really, this is how we look at these. We look at them in different wells in comparison to the reference. And so what this looks like when we actually get the test back is you can see that in particular in this, so you can see here that there's some missing material in this particular individual, and it's important to talk about what we can detect with this type of testing. So first of all, the missing or extra pieces have to be a certain size for us to successfully detect them. It's also really important to note that we can't say anything about structure. So all we can say is if there's a missing or extra part, but not much more about where that missing or extra part would be located. We need additional testing to tell us that. And then the other thing, and this is really important if we're talking about recurrence risk, or we often get questions about whether we should do familial testing or parent testing. And sometimes there can be something called a balanced translocation where 2 parts of chromosomes have sort of played do-si-do and switch it parts. And when we see that, that can indicate an increased risk for some of these chromosomal differences, and we cannot tell that from a chromosomal microarray. So it's important that testing is getting better and better, and we're doing more and more genetic testing, but often, when we see individuals with delays in development or concerns about disabilities, we'll often start with this particular test. So it's our most common genetic test that we send when we see someone with a diagnosis of being behind in their developmental milestones. And so how do these changes in copy number variants, so these deletions or duplications, occur? And the way that these occur is that on each end, so you can see here there's a flanking end, there are these errors indicate that there's repetitive regions. So these are highly repetitive regions, and the concern is that if these regions don't align well, which can happen because they're very similar to each other, then when crossing over occurs, what happens is you get a missing or extra part. So let's look at that. And it's very common for all of us to have these changes. So all of us have about 30 changes in our genes, maybe more. And these can be copy number variants. So we often each have missing or extra pieces. And part of the challenge here is determining which ones of these are what we call pathogenic or disease causing. So for example, one of the things we'll do, as you can kind of see depicted in this slide, is that we'll actually test a parent to see if we can determine if the specific change is inherited from a parent. that can be challenging when we're talking about some of the chromosomes that we're talking about in this room, like chromosome 15. But in general, we tend to use and try to understand if something's inherited to know if it would be causing a problem. And this would be things that are present in the exact same way in the child as they are in the parent. So this is different from what I was just talking about, where there's translocation, where the parent would have all of their material and wouldn't be missing any material, versus these inherited missing or extra parts of chromosomes, which may be what we call benign, meaning that they might make a family unique, but they don't actually cause any problems for learning or development or any other part of the body. So we're going to talk a little bit about mechanism of copy number variation, and you couldn't have more different chromosomal changes in this room. So you're going to hear about pretty much all the different changes that you could see in genetics. So the most common mechanism by which we see these copy number variants is something called non-allelic homologous recombination. So this is what I described in that. So you can see here that there's these regions that are what we call homologous, meaning that they have very similar repetitive regions within those regions. And what happens in those regions is something called crossing over. And the body is very dependent on a proper alignment of those changes. And probably easier to do this on this side. So you can see that if crossing over occurs appropriately, you would have the green crossover with the green and the red crossover with the green. And this occurs in something called meiosis 1, which is the first kind of transition from going from a germ cell to going from a cell in your body to what we call a germ cell, where you have one copy of every single chromosome that could be passed on from mom or dad in an egg or sperm. So you can see here, if that doesn't occur appropriately, you end up with two potential results here. One is the duplication and one is a deletion. So basically, we want chromosomes to align appropriately to one another. And if they don't align appropriately when they break apart, you can end up with a missing or extra piece. So inversion, and the reason I'm mentioning this is that this is very relevant to chromosome 8p. So this is a similar mechanism, but what you see here is in one copy, you can have overlap of the homologous regions within the same copy. So within the same allele or copy that's either from mom or dad. And so when the crossing over occurs, you get a break. So instead of having these exons, which are what are included in our genes, so our chromosome copies carry copies of our genes. And so when you get this crossing over, you get what we call this inversion, where you can have either BCD with on the same side of the chromosome, not crossing what we call the middle of the chromosome or the centromere, have this inversion where you get DCB, or you can have something that includes the centromere itself, or that central part that connects the short and the long arm of the chromosome, and you can get an inversion that crosses that center. And this type of inversion, the pericentric inversion, tends to be more likely to cause a problem because it's crossing over that center. So when we apply this to 8p, where this is most relevant, there are flanking regions, so those highly repetitive regions on both sides of 8p, and these are genes that are really important for how you smell, so they're olfactory genes. And so when you get this inversion that occurs, or you can get a deletion or duplication as well, it's really caused by the misalignment of those particular genes. And hopefully you'll get more detail, but if there's more questions about specifics, then I'm happy to talk with anybody after. So ring 14, we know that there's a ring formation. And we look at rings, there's two different mechanisms by which rings can occur. One of them is that there's a breakage at each end of the chromosome. So you have a short arm and a long arm of the chromosome. Here's that centromere that we were talking about with an inversion. You can have a breakage at each end of the chromosome, and then it can become unstable at those ends and form a ring. The other thing that can happen is just having, so your telomeres are at the end of your chromosomes and are very important for how we continue to replicate in our cells. And so you can also have these telomeres become unstable and form a ring. It's really important that you tend not to have missing material when you have this telomere dysfunction versus the double strand breaks, you can have deleted material as you see here. So in a large series of individuals with ring 14, it tends to be this telomere dysfunction that's more common in ring 14 based on what's available in the literature, which is relatively little. But in 21 of 27 individuals, I said that backwards. So in 6 of 27, it was that situation where there weren't missing pieces. And in 21 of 27, they had those missing pieces. So I apologize, I said that backwards. So double strand breaks actually tends to be the more common mechanism in ring 14. So the recurrence risk of these chromosomal differences actually is quite low. There's a few things that can increase the risk of recurrence, and I think these are really important to know about for families. So you can have an inherited change from a parent, and this is really important for duplication 15q because parent of origin is very important. And we're going to talk about that in the specific session for 15q. So you can have those as high as a 50% recurrence risk if something's inherited from a parent. There's also something called gonadal mosaicism, where you can have some of your egg cells or some of your sperm cells that carry that particular change, and it can be present in a subpopulation. So it's possible that when you get testing, it could be completely normal, and then in less than 1%. So very rarely do we see situations in which you can have gonadal mosaicism, or that subpopulation, one of the eggs or sperm gets passed on more than once. And then the last one is that situation of a translocation, as I mentioned. So you can have, so this is a nice depiction of, you have all of your material. So you would never have any symptoms from this. There are some rare instances where there could be an interruption of a gene at the point at which that you have that mixed up piece. But in general, most individuals have no idea that they could have a balanced translocation. They're completely healthy. They've never had any medical problems from it. And in that case, what you can see is that, say chromosome 15 has linked on to part of chromosome 20 and part of chromosome 20 is on chromosome 15, you can imagine that anytime you pass on those, what we call derivative chromosomes, you can imagine that you have an increased risk of passing on the one that has either the missing or extra part to predispose to have a child with either that missing or extra part. So genetics, the field of genetics is actually, I think, changing because we've gotten better at diagnosing individuals. And now I think we're moving more towards a role of management, both precision medicine, targeted therapies, but also just coordination of care and management. And so what I tend to do is continue to follow individuals with differences in their chromosomes or neurodevelopmental disabilities. And we have a lot of things that we can actually offer. So making sure we're coordinating care, thinking about all the risks, making sure we're monitoring labs, making sure that we're thinking about the family as a whole. A lot of times we also do something called genotype-phenotype correlation. So, and you may have heard about that a little already, but basically what we're thinking about is the particular change that each individual has in the chromosome, which can vary, and what symptoms tend to be more commonly associated with that particular change. And so the other thing we do is a lot of talking with the primary care physician, making sure we're not missing anything that could be involved. And then again, we're really interested in making sure that we're thinking about targeted therapies and how do drugs that already exist how can they be repurposed to maybe help a particular genetic change? But then also thinking about how do we design a targeted therapy for the different specific genetic change that your child has. And so I just want to end with emphasizing, so this is not any of these one disorders because I didn't want to pick one, but you can see that this is a chromosomal disorder that has to do with the 15 chromosome. And you can see that many different organ systems are involved. And so we think it's really important to make sure that you have someone who's thinking about all the possibilities and making sure they're not missing anything. And the other thing that we kind of provide is this, a lot of family support, access to resources, making sure that we're taking care of the whole family. A lot of times there can be increased anxiety, and we're doing a lot of research about sibling health at Children's Hospital Colorado. And then natural history and understanding natural history and outcome measures is obviously very important, especially going forward and thinking about precision medicine. So I could not do this without my amazing team at Children's Hospital Colorado. I'm so thankful for them. We really try to know everything there is to know. A lot of times it's the parents that are teaching us, but every single clinic, I think I learned something new about duplication 15q, or anytime I see any chromosomal abnormalities, 8p, we see quite a few, ring 14, we're always thinking about learning and passing on anything we learn from all of you to everyone that we're seeing. So thank you for that.
So we don't have any questions on the app, but I see a lot of eager people taking pictures of the presentation. So if you guys just wanted to raise your hand, I can pass a mic to you. Do you see a hand?
No, he was a cutie in the front. That one.
Hi, I'm Evan. I'm a father of an 8P little girl. And I did have a question. This is such a layman's question, but I'm sure lots of parents in this room maybe have wondered about us the same thing. But so we've been told that the chromosomal difference was de novo. And we're thinking about having more children. And there's that like fear that maybe they're missing something, right? What would you, I guess, can you speak to any of that? The faith we could have in our geneticist who told us that, the lab, the uncertainty, thank you.
Yeah, absolutely. And have you all had any genetic testing?
We both did, and that's when they told us it was de novo.
Okay. Yeah, so if I always recommend doing karyotypes for parents, especially if there's some anxiety, because it can be helpful to look at them just to make sure that there's nothing that we're missing. So that's the one where we look under the microscope and make sure that the structure of the chromosomes look normal, because I think that that could be really helpful. And if there can be some signs, though, that a translocation exists, so not all geneticists would recommend that. If you look at the specific chromosomal testing that's already been done, if they've looked at structure and they don't see anything concerning on the child, they sometimes don't recommend that. But that's just something that I've started recommending because I find it's helpful for also anxiety of having another child. The karyotype, to look for a balanced translocation. Mm-hmm. Absolutely. Really fast. Nice. Can I go back to my picture of the chromosomes?
Yeah.
Oh, here we go. Okay. So the answer to that is no. Some chromosomes are more gene rich than other chromosomes. So the way that we look at that, and it has nothing to do with the numbering is actually based on the size of the chromosomes. But what you can see here is that this is called Giemsa staining, and you can see that there's these regions that are light and these regions that are dark. And the light regions tend to have more gene-rich regions or active genes in them. And so sometimes you can have a huge missing piece or a huge duplicated piece and not have any symptoms because it's a relatively gene-poor region. But it doesn't, so that's what I would say is that depending on the chromosome, like chromosome 1 is relatively gene poor, and we can see kids who have really large extra parts of chromosome 1 or deleted parts and not have any symptoms from it.
So when my son was diagnosed with 15q, my husband and I opted not to get tested. But if we get tested now, can that like benefit anything for him?
Good question. So there are some additional steps of testing in 15q that we recommend for duplication or deletion actually to determine definitively whether that extra part is from mom. Because we do have cases where the extra part is from dad. And what we're learning is that those cases are not always asymptomatic. So sometimes they have milder symptoms. So I don't necessarily, doesn't mean you guys have to have testing, but you can do also a test called a FISH test to look at structure of chromosome 15 to look to see if the extra part is in the child is interstitial, which means within the chromosome itself, or if it's an extra part of a chromosome called a marker chromosome or isodicentric.
Okay, so his is also translocated. Does that change anything? So like his extra, he was isodicentric, and his extra is on top of his 14th chromosome.
So that we could definitely talk offline.
Okay.
And I'm happy to look at it, but something like that could indicate that there might be a slightly increased risk of recurrence risk. And so I would, depending on where you are in your planning, think about that. But I'm happy to talk more about it.
Great, thank you.
Yeah. Okay. So someone asked, is there a value in retesting after 15 to 20 years to see if the size of the duplication decreased? So there isn't a change in size of the duplication over time, but we've gotten better at understanding the breakpoints and our technology has gotten better at understanding the size. So I don't necessarily recommend retesting if you have the information, you know, the structural information, you know, whether for duplication 15q, if whether you know if it's from mom or dad. I don't necessarily recommend retesting. But the technology has changed. You may be able to get more information from the testing available. In a known inherited unbalanced translocation, what is your general advice for sibling testing before the siblings are at an age of consent or understand? So I wouldn't recommend doing any testing in the sibling unless parents have had testing that indicate that there could be one of those balanced translocations or duplication in the family to try to understand if there's an increased risk in the sibling. So, and I would recommend if the sibling is completely neurotypical that you wouldn't do any testing till they can make that decision themselves. There's a couple more coming in. Is IDIC form of DUP15Q always de novo? So from what we understand, most of the time it's de novo. I have had a few cases where it has not been new in the child or what we call de novo. So it's still worth getting the full battery of testing. And then my daughter is isodicentric and has a tetrasomy. Does tetrasomy necessarily have more severe disabilities? So when we've done studies to look at the size of the missing part of, or the extra part in this case of chromosome 15, It does seem to be that the more extra copies there are, the more severe the clinical picture is, but that's not always true. So we can see individuals who have a tetrasomy who are very mild as well. So the spectrum is very wide, and it doesn't seem to be solely represented by the chromosome itself. So there may be other factors that play a role that we haven't even identified yet. Think I got all the questions, but I'm always happy to chat offline with anybody, and I'm always happy to give my email too.
Absolutely. Thank you.